everybody. Welcome to a new expert talk. These things happen because you happen, because we're here for you and we know that you're here for us. So, one special announcement. We get together, we spend an hour and a half, we do a lot, but the rest of the day, remember about the Pathfinders. There's 16 of us. You can just go everythingals.org/pathfinders, and you will find our home away from home. And I hope you make it yours too. And with that, I'll turn it over to you, Indu.
Hello, everybody. Nice to have everyone here. And I am so thrilled to actually welcome Dr. Levy and also Dr. Olga here. And Christian, do you want to do the introduction, please?
Sure thing. So thank you, everybody, as Indu and McFinn has welcomed us. Tonight's a special night where we get to have a double program for members of our pre-competitive industry consortium, companies that not only are working on their own clinical trials and investigations in ALS, but also are incredible supporters of the work we do here and believers in what we do. I can honestly say that both Oren and Olga are incredible contributors in their own rights, as they have been consistent advisors to the ongoing work we do, including advising us on, what's going to not only make for data collection that can help clinical trials, but also things that can help us get across the finish line, getting drugs approved, not only in the US, but abroad as well around the world. So with that, I'm really excited to have them both be able to join us tonight to present updates on their respective companies' work in ALS. Dr. Oren Levy is going to go first. I'm going to introduce him, and then I'll also introduce Dr. Olga Uspenskaya-Cadoz really quickly too. Oren Levy is medical director for early clinical development at Regeneron Pharmaceuticals, where he leads initiatives involving digital biomarkers, physiological signal-based assessments, and an innovative early phase trial design in neurodegenerative diseases. He's a skilled physician scientist, and Dr. Levy applies his dual training to integrate advanced digital tools and patients-generated data into clinical research. He's published studies on remote respiratory monitoring in ALS and mobile gait analysis in Parkinson's disease, reflecting his commitment to objective, scalable trial endpoints. His work is dedicated to enhancing precision and efficiency in early clinical development through technology-driven approaches. Also joining us is Dr. Olga Uspenskaya-Cadoz, and Dr. Olga was appointed as Chief Medical Officer at Vectory Therapeutics in April of this year. Dr. Uspenskaya brings over 2 decades of expertise in neurology, gene therapy, and clinical development to Vectory. Within Vectory, she leads clinical and regulatory strategy as the company advances its pipeline of vectorized antibody therapies for neurodegenerative diseases. Before joining Vectory, Dr. Uspenskaya-Cadoz was Vice President of Medical Gene Therapy at Eli Lilly, where she oversaw clinical development, pharmacovigilance, and patient advocacy for CNS and lysosomal storage genetic medicines. She also played a key role in shaping the company's ALS and rare disease strategy. Previously, she served as Vice President of Clinical Development at Prevail Therapeutics, where she contributed to the design of novel gene therapy programs for neurodegenerative disorders, which was eventually acquired for their great work by Lilly. Dr. Uspenskaya-Cadoz holds an MD and PhD in neurosciences from First Moscow State Medical University, with additional research training at Ludwig Maximilians University in Munich and Sorbonne University of Paris. She's authored over 20 peer-reviewed publications and is presently presented extensively at international scientific conferences. We welcome both of them. And as I mentioned, Dr. Levy is going to go first with his updates from Regeneron. So we will turn it over to you.
Okay, can you guys see my presentation?
Yes.
Great, thanks. Thanks, Christian. Thanks, Indu, for the opportunity to speak this evening. It's a real pleasure. Quickly, my disclosures. I think as Christian mentioned, my background is as a neurologist. Before I saw patients mainly with Parkinson's disease at Columbia before joining Regeneron about 5 years ago. It's been a real kind of privilege to be able to be part of this community and really learn from the community about ALS and try and bring that perspective and that knowledge to the work that Regeneron is doing. A brief background about Regeneron Pharmaceuticals. It's been around for almost 40 years, founded by two physician scientists who are still running the company, and actually founded initially to try and develop a treatment for ALS back in the late 80s, early 90s. They did some of the early work related to neurotrophic factors in ALS. Unfortunately, that didn't pan out. They've stuck with it. Really built an incredible company that has really spread far and wide beyond neuroscience to develop many drugs across many different diseases. But the passion and interest in ALS has been a constant. And we're really excited to be back in the clinic and tell you about some more of the work that we're doing to try and better treat and understand ALS. I think Regeneron is probably best known for its expertise in monoclonal antibodies. That's what most of our approved drugs are. But I think we realized a few years ago that while antibodies are fantastic tools, they don't do everything, and we really needed to expand our toolkit and our platforms to be able to treat a wider variety of diseases, including brain diseases. And that was really the driver that led us to form this collaboration with another company, Alnylam Pharmaceuticals, to help develop a novel class of gene therapies or nucleic acid therapies called siRNAs, or small interfering RNAs. For the treatment of neurological diseases, and that's part of the story I'll be telling you today. We really try at Regeneron to follow the science. We really want to understand, what is the best scientific argument behind a treatment, and we follow that where it leads us. If it leads us to a very, very small and niche indication, so be it. We really want to try to let the science speak. And we also want to let the patient speak. Everything we do is really focused on the patients that we're trying to help. So today I'm going to tell you about three broad initiatives that we're undertaking to advance treatments in ALS. The first is a novel therapeutic we're developing to treat ALS that's caused by mutations in the SOD1 gene. Again, this is part of the siRNA platform that we're developing in collaboration with Alnylam. The second is work that we're doing to really just better understand the disease. And this is really through membership in different consortia that are trying to basically do research to better understand biological basis and disease progression in ALS. And then the last point is really partnering with the community, really trying to work with patients to understand how we can do our work better and what we can learn from them in order to really make the most impact with the work we're doing. And before I go on, I really just want to acknowledge that while I'm here talking to you, this really represents work done by a very, very broad team. There's an enormous team that goes into bringing a therapy through the lab and into the clinic and actually getting it to patients. And there's an amazing team I work with in patient advocacy, medical affairs, that helps do the work in terms of working with and partnering with the ALS community. So this is really work that we all do together. Okay, first story. The work that we're doing with Alnylam to develop siRNAs is really kind of based on very fundamental cell biology. And really what it is that there are messages that your cell makes that basically code for proteins. In the case of SOD1 ALS, SOD1 is the protein that we want to turn off. People with SOD1 ALS have mutations that cause their SOD1 protein to aggregate and become toxic. So what we're trying to do is essentially decrease the levels of SOD1. And we can basically take advantage of the genetic code and of the cellular machinery that already exists to try and regulate different genetic messages, to try and very specifically and in a very targeted way decrease the levels of the SOD1 protein. Companies can take kind of different approaches that try and do the same thing. So there are two kind of platforms or approaches that people are using to do this. One is called antisense oligonucleotides, or ASOs. So those of you who are familiar with tofersen or Qalsody for SOD1 ALS, that's an antisense oligonucleotide. We have kind of a similar but distinct approach or platform, which again is called siRNAs, that try and do the same thing, basically reduce the level of SOD1 and do it in a very, very targeted way. Essentially, what we do is we take advantage of this cellular machinery that exists to really help regulate messages in cells. The siRNA kind of hops onto that machinery and acts as a template that the message for SOD1 can bind to. And when it's in there, the cellular machinery basically cuts it and degrades it. And this can happen over and over and over again. So we can really, in a very strong way, reduce the levels of SOD1 message and therefore reduce the levels of SOD1 protein that are driving the ALS in patients with these SOD1 mutations. Now, these siRNAs are fantastic. Unfortunately, they can't get into the cell on their own. We need to really attach something to them. We call that a ligand. And for the brain platform, this ligand is called C16. This C16 is basically stuck onto the siRNA and basically gets it into cells in the brain that we want to target. As I said before, this was a novel collaboration and really a novel approach. The first program that our companies worked on together that went into clinic back in 2022 was a program that was targeting the APP, or amyloid precursor protein. This is the protein that's involved in the development of Alzheimer's disease and the generation of amyloid plaques. This was a big moment for our companies because this was the first time that we're aware of that an siRNA was administered to a patient to try and target a brain-encoded protein. And as I'll show you, the results were positive and really kind of established that we can use this platform to specifically target proteins in the brain and in the spinal cord. So the second program that we brought into the clinic is the program I'll tell you a little more about, which is the program that targets SOD1. So again, this is for patients who have a SOD1 mutation that leads to ALS. We've done a lot of work in cell models, in animal systems to show that this siRNA can very strongly reduce the levels of SOD1, which is exactly what we want to do. So I mentioned the first trial that we did with the ALN-APP molecule, and I'm showing you results from this. I'm going to talk you through it mainly because, again, this is the first time we tried this in humans, and we were really excited when we saw this data. So this was the first trial that was ever done with this molecule. It was in patients who had Alzheimer's disease. They were given a single dose of the drug. The drug has to be administered through a spinal tap, similar to the way tofersen is. And then these patients get additional spinal taps over time. And we measure the levels of an APP protein in their spinal fluid to tell us, is the siRNA actually reducing the protein that we're trying to target? If you see the red line, that's basically what happens to patients who were given the placebo treatment. And basically their levels are nice and steady. And the first dose we tried was in blue, the 25 milligrams, also pretty steady. The second dose we tried was 75 milligrams in the yellow. And what we saw was a very rapid, a very strong fall or reduction in the levels of the APP-derived protein in spinal fluid down to about 75, 80% lower than where it started, and lasted for an incredibly long amount of time. Again, these patients got a single dose of the drug. And at 6 months, APP levels are still strongly reduced, and it's very gradually recovering. But even out to 10 months, there's still substantial reduction. So again, this was really surprising, encouraging. The next dose we tried, we actually lowered it a little bit and tried 50 milligrams in purple. And you can see we're still getting really strong reduction, and it's wearing off a little bit earlier. So again, proof that the platform that we had been studying for many, many years in animal systems actually does what we wanted it to do in people, which is strongly reduce the levels of a target protein and do that for a very, very long period of time. You can imagine these drugs have to be administered by spinal tap. So the ability to be able to only have to administer the drug twice a year, maybe even less frequently than that, is really, really appealing. So as I said, the second program was in SOD1. The animal and cell experiments told us that this is really strongly reducing SOD1 protein. And we've actually moved forward and we've started doing a clinical trial with this in patients with SOD1 ALS. We tried to be really thoughtful when we designed this study to make it as patient-friendly as possible. I'll talk about this a little later. We convened a patient advisory board who was really, really helpful for us as we thought about how to design this study. In some ways, this is kind of what a typical study looks like for a drug when you first put it in a person, in the sense that you give a dose, you monitor the patients, and then you give a little bit of a higher dose, monitor the patients, give another higher dose, and monitor the patients. The adjustments we made were, one, we reasoned that people with SOD1 ALS, especially if they were in regions that have access to tofersen, would really not be able to tolerate or sign up for a trial that involved a lengthy placebo period. So what we did was we shortened the placebo period to 4 weeks, and basically made it so that anybody who entered the study, by 4 weeks into the study, they knew that they were going to be getting active treatment with the active drug. And we thought that was really kind of important from the patient perspective. And we didn't think we were sacrificing anything in terms of what we were learning from the study, in terms of understanding the drug and understanding what it was doing. We also realized that SOD1 ALS is really, really rare. It only accounts probably for about 2% of all ALS cases. So there aren't a lot of patients out there. So we really wanted to make the most out of every patient, learn as much as we can, and really kind of get to the doses that had the best chance of helping patients as quickly as possible. For that reason, each dose level that we're studying, we start off with 3 patients. And we follow those 3 patients, and we make a decision about whether or not we can try the next higher dose based on those 3 patients. So again, that really allows us to, as efficiently, and we think as safely as possible, get to doses that have the best chance of helping patients. Now, once we kind of go through that process of getting up to a higher dose, we can then go back and say, oh, this dose that we tried before, we want to learn a little bit more about that one. We're going to add more patients to that group. So we built in that level of flexibility. And then the last thing we reasoned was we really wanted to offer patients more than a single dose of the drug. We wanted to offer patients multiple doses over a long period of time. So in this study, we committed to treating patients for 2 years, and that they would get doses every 6 months and potentially even less frequently over that 2-year period. So this is, again, some of the elements of this study that we really try to think about, what will help us learn the most about the drug that we can in a way that is kind of most patient-friendly and most patient-centric. This trial is ongoing now, and we have the reference there for the clinicaltrials.gov site to learn a little bit more about it. So I'm going to shift gears now and talk about some of the work we're doing to really understand the basics of ALS. And this work has really gone or happened through different teams and consortia that we've joined as sponsors and funding partners. One of our earliest efforts was a Canadian effort called Capture ALS. This was essentially a natural history observational study across multiple centers in Canada to collect data with a specific focus on imaging in ALS patients, a really rewarding experience for us. We leveraged that to basically do a sub-study, so within a small group of those patients, to study a digital insole that you can put in your shoe to measure gait features. So we're, in the process of analyzing the data from that small pilot study. Some of you may have heard of the AMP-ALS program that's being led by the NIH and multiple other partners. This is, again, in a broader way to try and collect and disseminate as much data as possible about the natural history of ALS. And we're really happy to be a part of that effort. And then the bottom one is a scrappy little outfit that you guys may have heard of that we've been working with for the past year or so. Our experience working with EverythingALS has been fantastic. We really value, what they bring, their approach to data science, their approach to really bringing patients in. So it's been a fantastic experience working with them. And then this is my last slide that talks about some of the work we're doing with ALS patient community. We really reasoned that we could learn a lot from patients. If we're able to talk with patients, we can incorporate what we learn into the way we do our clinical trials, into some of the outreach that we do to really be able to engage with patients. So one of our earlier efforts, as I mentioned, was a patient advisory board that we did as we were preparing to start our first in human study. We really learned a lot about things that are important to patients, generally speaking, things like education, mental health support, working with sites, getting very clear communication, the value of these support groups and programs to build a sense of community and camaraderie with the patients. And then even kind of more nitty-gritty procedural things about how annoying it is to have to do an overnight stay at a site and to really be thoughtful about when we ask patients to do that, if we really need to do it, or maybe we don't have to do that. Really, really valuable. A parallel effort we did was we did a literature review to really understand some of the best practices that different patient advocacy groups were doing around the world. And this was work that we presented a month or two ago at the PACTAL's regional meeting for ALS, and just highlighting three of the regional initiatives that we characterized. One in Taiwan was looking at an eye tracking communication device for people with advanced disease who needed help with communication. I think one really nice thing about that effort was that it really focused on accessible, affordable technology that would be easy for patients to be able to gain access to. A second effort was undertaken in Japan. This was a survey to explore brain-machine interface and attitudes about that. This was really interesting. In Japan, there's a more negative attitude towards ventilatory support. It's really seen as burdensome, especially to patients' families. So it was interesting to see that there wasn't so much that negative association with brain-machine interface and maybe more openness to using it. And then the last effort was in Australia. Again, this was a survey to look at access, acceptance, and use of non-invasive ventilation across the country. And I think one very interesting insight there was that they found that patients who were in rural areas and patients who were women were both much less likely to be offered non-invasive ventilation and much less likely to accept it, even if it was offered. Clearly a critical insight if you're trying to expand the use of this really important approach to as many patients as possible. So again, today I told you about kind of three categories or broad efforts that we're doing to develop a novel treatment for a genetic form of ALS, our work that we're doing with partners to better understand the disease, and then the work that we're doing with patient advocacy groups, all with kind of the end goal of improving the lives of patients with ALS. So thank you for your attention, and thanks for the opportunity to speak.
Thank you, Dr. Levy. And by all means, if you have any questions for Dr. Levy as we go along, you can continue to drop them in the chat. We're going to be continuing with the gene therapy themes and precision medicine themes now, as I help Dr. Olga Uspenskaya-Cadoz present on behalf of Vectory Therapeutics.
Thank you, Christian. while you are pulling the slides, I just wanted to say good afternoon to everybody, and thanks for inviting me to provide an update on what we are doing, to introduce our company as well to most of you. Maybe you never heard about what Vectory is, so I will try to introduce our platform and provide some information on our lead asset we are developing for ALS. So let's go to the next slide. So we are headquartered in Europe, in Amsterdam, and we also have some presence in Boston. We have an office in Boston, and we are a biotechnology company founded in 2020 with a mission to develop vectorized antibody treatments for people with neurodegenerative diseases, enabling longer, better lives, and reversing the course of the neurodegeneration. The company was founded by a physician scientist who is a true veteran in the biotech, who developed actually multiple treatments which are on the market already, and one of them, which recently provided some very exciting results, is a treatment for Huntington disease, a treatment which is being studied by uniQure. Let's go to the next slide. Yeah, and you obviously know that neurodegenerative diseases affect millions of people worldwide, and this number is growing, unfortunately. And despite some approved treatments, both in ALS and especially in Alzheimer's disease, there is still an immense need for new therapies that treat the underlying causes of neurodegeneration. So our current pipeline programs are really heavily focused on amyotrophic lateral sclerosis, followed by Huntington disease, Alzheimer's disease, and frontotemporal dementia. Next slide. So at Vectory, we are experts in the field of intracellular and therapeutic antibody design and adeno-associated viral vector development. So we really take the best of both worlds in intrabody development and vector development to be sure that we are delivering the therapeutic antibodies where they reach their target. So our technology overcomes the limitations of the current therapeutic approaches, including delivery, durability, and target accessibility. Indeed, we are able to deliver the therapeutic intrabodies directly inside the neuronal cells, targeting toxic, pathologically misfolded proteins with high precision while preserving the normal protein functioning. Next slide. Let me just spend now a few minutes on our lead asset, which we call VTX002, which is a vectorized antibody targeting TDP-43 in ALS. And I'm doing this not only to introduce our approach, but also to set up the scene for some additional discussions on the particularities of clinical trial execution with novel therapeutic modalities, and what we need to do to make them a success in ALS. Next slide. So I'm not going to spend a lot of time here. We all know that ALS is a terrible disease. It is universally fatal. But what is important here is that in approximately 97% of cases, the underlying driver of ALS is TDP-43 pathology. And this is what we are trying to address with our lead asset, VTX002. Next slide. So what TDP-43 is? So it is really a crucial nuclear protein which is encoded by TAR-DBD gene, and it is essential for the healthy functioning of motor neurons. It acts as a nuclear transcriptional repressor and is involved in RNA metabolism, including transcription and splicing of mRNA, maintaining RNA stability, and axonal transport in motor neurons. The key pathological hallmark of ALS is the mislocalization of TDP-43 from the nucleus to the cytoplasm and the subsequent formation of aggregated TDP-43 inclusions in upper and lower motor neurons that subsequently may undergo post-transcriptional modifications, including phosphorylation. This leads to the loss of function in the nucleus and a toxic gain of function in the cytoplasm. And this loss of nuclear TDP-43 function is particularly significant as it disrupts the protein's autoregulatory feedback loop. Normally, TDP-43 binds to its own mRNA, targeting it for degradation through a nonsense-mediated decay pathway. And this process is crucial for maintaining the precise levels of TDP-43 within the cell. But unfortunately, when TDP-43 is pathologically sequestered in the cytoplasm, this autoregulatory mechanism fails, leading to an uncontrolled accumulation of TDP-43 mRNA and subsequent overproduction of dysfunctional protein. And so this, unfortunately, perpetuates the toxic cycle of the TDP-43 aggregation and loss of function in the nucleus. Next slide. The investigational product, VTX002, we are developing is a recombinant adeno-associated virus-mediated gene therapy vector consisting of an AAV5 capsid and an expression cassette encoding a humanized anti-TDP-43 antibody, which targets and binds to the misfolded and aggregated forms of the cytoplasmatic TDP-43. This construct was designed to reduce the pathological aggregates in the cytoplasm and restore the normal TDP-43 functions, such as cryptic splicing, which is associated with ALS pathology and TDP-43.
Network coverage and customer satisfaction.
And so this product underwent a really extensive preclinical testing, both to demonstrate efficacy, but also to ensure that it is safe. We conducted a very robust preclinical testing in non-human primates to be able to move forward with planning of our clinical trial. So this preclinical testing is now complete, and we are in the process of getting a regulatory review of our application to conduct a clinical trial. Next slide. So I'm going to talk today a little bit about what we saw in the preclinical setting, and I hope that maybe early next year we will be able to reconvene and present and talk about our planned clinical trial. We are hopeful to start patient dosing actually early next year. And the study will be conducted in the United States and also in Europe. So the pharmacology of VTX002 has been characterized in human neuronal models, and these studies demonstrated that the therapeutic efficacy of VTX002 in ALS animal models is really very compelling. We demonstrated that the target engagement was a reduction of TDP-43 aggregation and correction on the next slide of loss of function of TDP-43 by correcting cryptic mRNAs and ALS transcriptome. Next slide, please. Yeah, so you probably heard about this cryptic splicing events. There are therapeutic strategies which are addressing specific proteins like stathmin-2 or ONC38. They are part of TDP-43 pathway. And what we were able to see in the preclinical testing of VTX2 is that administration of a vectorized antibody allows to correct multiple cryptic splicing events, not just one. Let's go to the next slide. So I will also, like Oren, will switch the gears and talk about the particularities of clinical trial execution with this novel therapeutic modalities. They are much more complex than a standard ALS clinical trial, and they require a really close and early engagement of drug developers with patient advocacy groups like EverythingALS. So we actually, as well, in the preparation of our future clinical trial, conducted patient advisory boards to understand your opinion on our plan and to improve our clinical trial design, decrease the burden, and ensure that what we are doing is truly meaningful to the patients. Next slide, please. So one of the important considerations when conducting these kind of clinical trials is that we know from other neurodegenerative disorders that it is really of utmost importance to be able to treat the patients as early as we can. And unfortunately, ALS is still being diagnosed way too late after symptom onset. So there are a lot of efforts in the scientific community to shift this diagnostic paradigm to the left to enable an earlier intervention. And some of these considerations are being implemented in our planned clinical trial to be able to start treatment as early as possible to increase the probability of treatment success. Next slide. There are some additional clinical development considerations for genetic interventions in ALS. Well, first is the choice of the right clinical endpoint. There may be a regulatory precedence and natural history trajectories available for larger patient population, but may be missing or incomplete for a genetically defined patient subpopulation. May be also relevant to the subset of patients at a specific stage of the disease. So this is something which is important to keep in mind. The choice of right surrogate endpoints, the regulators always ask whether the biomarkers which clinical developers incorporate in their clinical protocols correlate with clinical progression. And is the change on this biomarker clinically meaningful? Whether the assays are validated and available. And also we want to understand early on, this is something we are doing as well as part of preparation of our clinical trial. We're actually conducting ourselves a collaboration with Utrecht Medical Center where we get access to the natural history data from the patients suffering from ALS and their biosamples to understand the correlation of the readouts of some novel biomarkers across TDP-43 pathway in the CSF and the periphery. Of course, if there is a strong correlation, there is an obvious preference to decrease patient burden and to test them in the periphery versus CSF sampling through lumbar puncture. Heterogeneity of patient population is a key consideration in ALS. So what is the right window for intervention is another question. Can we later broaden our patient population? This is the question we'll have to answer as well. What is the appropriate timeline to assess the key endpoints? We have some lessons learned from previous ALS trials. And this is something we also keeping in mind when designing our clinical trial. Also, ALS what was great about ALS that there are a lot of attention from biotech and pharma companies. So there are a lot of trials which are ongoing, and hopefully there will be new drugs approved soon. So when conducting clinical trial, there may be a situation when the standard of care is evolving during the conduct of your clinical trial. So this is something one should incorporate in the thinking as well when analyzing the data later on. And also with this novel treatment modalities, oftentimes we have to go with somewhat invasive route of administration. It can be a delivery through a lumbar puncture, or like in our case, it is a delivery via cisternal puncture. It is one and done treatment, but it requires a cisternal puncture to deliver the drug. And there is still a high likelihood that for a full registration, there may be a need to conduct a very complex sham control study with complex blinding procedures, which require a lot of logistics, in-depth site training, but also which may be challenging to be accepted by the patients and may be not seen as highly ethical. So this is also an ongoing discussion the field has in terms of how to conduct this registrational studies with less straightforward routes of administration. Next slide, please. So this is just the picture demonstrating the complexity of ALS targeting gene therapy trials. On the top of all ALS trial complexities, there are much more specialty labs. The patient will have to get much more biomarker sampling. There are also additional biomarkers to understand the immunogenicity profile of a gene therapy asset, for example. There are additional regulatory and startup obstacles one needs to overcome to get the trial up and running. It takes longer time for these trials to be initiated. And the manufacturing complexity of this kind of treatment modalities is really immense. Next slide. And then when the study starts, there is a need of much more intense patient monitoring to really understand the safety profile of, especially when it comes to AAV-based gene therapy targeting neurodegeneration. So it is critical that these studies are conducted by highly experienced neurologists, which are able to disentangle potential safety signals coming from underlying disease progression versus those which may be related to the investigational treatment. There may be some potential drug delivery procedure risks, but also some additional safety risks coming from gene therapy per se. Next slide. Yeah, and then of course biomarker development is critical not only for genetic medicine in ALS, but in general for ALS clinical development. And here the collaboration and natural history efforts are really paramount for success. We need to understand how the novel biomarkers behave in the natural history setting, how fast they change over time, whether they change over time at all, and whether they can be leveraged for the clinical development as a biomarker for patient selection, or whether it can be a biomarker which will help us to understand how our drug impacts the underlying disease progression. Next slide. And so there are multiple types of biomarkers. There may be a diagnostic biomarker, prognostic biomarker, pharmacodynamic or safety biomarker. And actually, we are lucky enough in the ALS field that we have NFL, which actually ticks multiple boxes here and can serve in multiple contexts of use. We are obviously going to leverage NFL in our clinical trial, but we are working hard together with the larger scientific community on developing much broader biomarker strategy, and specifically working on getting hold of biomarkers which will help us understand target engagement with TDP-43 protein. Next slide. And so ideally, we would want to have a set of biomarkers helping us to understand both TDP-43 gain of function and loss of function. There is a lot of excitement about measuring TDP-43, for example, in extracellular vesicles. You can derive extracellular vesicles, which will be neuronally derived extracellular vesicles, so will help to really appreciate the levels of TDP-43 from the central nervous system. And the exciting part of extracellular vesicles is that they can be obtained from the periphery, so not from the CSF. But a lot of work still needed to better understand how the readouts overlap in ALS versus FTD, other neurodegenerative disorders, and even just healthy aging, and whether these levels increase over time or whether they plateau. These are still things we need to understand, and this is really natural history data which will help us, and academic collaborations which will help us to better understand the behavior of these novel biomarkers. So next slide. Yeah, I think I will just skip this one. Yeah, and then I will just summarize what I just presented by providing some topics for future success. And here again, I'd like to repeat that collaboration is really critical for success, that shared pre-competitive platforms for biomarker validation will be critical to understand both fluid biomarkers, but also imaging and digital endpoints. Academic-led natural history cohorts and biobanks can really help us de-risk early phase programs. There is a lot of opportunities for doing better job with joint platform trials and master protocols. The Healy platform is doing a lot of pioneering work here, but there are other platform trials which potentially could be fit for purpose for these genetic medicines with more invasive routes of administration. We also need to do much more collaborative work on delivery technologies. As I mentioned, our treatment will be delivered intrathecally through external puncture, but ideally we would want to have delivery technology not exposing patients to invasive procedures. And here there is, of course, as well an opportunity for collaboration with academia. And then we are doing all of this for patients. So early patient involvement in endpoint selection, trial design, and recruitment strategies are really critical. And not only in the beginning of the study, but as the study progresses, getting regular touch points with the patient groups to ensure that we continue to do the right thing, I think is really important. So with this, I think I'll stop. Thanks for the attention.
Thank you, Olga.
Thank you so much, Olga. All right. We have a few questions from the audience so far. Anusha is going to be joining us here to help moderate the questions along with Indu and myself. So. All right. Let's see. First question. I'll jump in on the first question. The first question was for Oren from your original presentation, Oren. What are off-target effects of siRNA when given intrathecally?
Yeah, great question. So part of what we do is and this is really Alnylam, they're the siRNA experts. Part of what they do when they select the exact sequence from the siRNA is that they basically comb through a database and try and pick a sequence or a part of the genetic message that is unique to that gene and not found in other genes. So that's kind of done at the computational level. We also do things like try and make sure that that region is relatively stable and constant so that it's the same across multiple people. And then we do a lot of experiments in cell systems and in animals to look to see, are there other messages that are being affected by the siRNA. So that's part of kind of the lab upfront process that goes into picking the exact siRNA that will eventually bring into the clinic. So I hope that answers the question.
Thank you.
Thank you. All right, the next question is, how much did and this is for Oren as well. How much did the rarity of SOND cases to enroll in your trial and the expected magnitude of therapeutic effect play into this unique trial design? For example, would something similar be rational in SALS, where a drug might be expected to have a smaller effect in a more heterogeneous population, and there are more subjects to enroll?
Yeah, I'd say it played a large part. So clearly, this is a rare population. I think the other thing that played an important part is that, we really benefited from the work that Biogen did as they were developing tofersen. So we really had a good understanding of the different outcomes that we were going to be using in our study. Specifically, how does SOD1 protein levels in the spinal fluid, are they stable over time? Do they go up? Do they go down? It turns out they're stable over time. And similarly with neurofilament light, or NFL, that Olga mentioned, we knew based on prior work that the levels of that are also pretty stable over the time course that we were looking at. So kind of with that knowledge in mind, we felt like we would be in a good position to understand the changes we were seeing and really be able to figure out, are they due to our drug or are they due to something else? So I think it was a combination of the rarity and also the prior work that had been done that gave us confidence that we could kind of figure out and learn what we needed to in the absence of a concurrent placebo control. And I think it's up for debate about whether or not this is something that's more broadly applicable to, non-genetic forms of ALS. Is a concurrent placebo control needed in a first-in-human study to understand the safety profile of a drug, if you really have a good idea of what to expect in terms of what happens to patients with ALS? Obviously, a lot depends on the specifics of the drug, and it's going to be case dependent. But I do think there are arguments to be made that in some, early stage programs that you can really kind of minimize the degree of concurrent placebo control that you have in the study.
Thank you. For next question, I have a question for Olga, and then we do have a couple of questions before we get too far into individual ones for both. Olga, how does VTX002 stop both sense and antisense dipeptide repeats?
Yeah, so, well, actually, I think that the question relates to the C9orf72 mutation carriers. It doesn't work on dipeptide repeats. It addresses the consequence of C9orf mutations, which is TDP-43 pathology. So indeed, VTX002 would be an appropriate treatment if it works for C9orf72 mutation carriers, and we are planning to include those in our clinical trial. So it's not necessarily working on dipeptide repeats. It works on the consequence of the mutations.
Thank you. That's a good clarification as well as far as who would be eligible for the trial as you prepare it. So thank you.
We are going to exclude patients who have SOD1 and FOS mutations because they do not develop TDP-43 pathology, but C9orf72 patients do develop TDP-43 pathology, so they will be part of the trial.
Thank you. Anush, you want to take the next one?
Sure. We can move to a question for the both of you. So could you both kind of comment on the difference between running ALS clinical trials in the US versus internationally? Are the clinics and the resources comparable? And kind of what do you think are the positives and negatives from an industry perspective? And then going forward, do you kind of expect more trials to be conducted outside of North America?
Well, I think it's different for
Olga,
SOD1
you go
versus,
first.
yeah, okay. Yeah, I think it's there are different considerations when running a clinical trial for a genetic subset of ALS where a standard of care is available. So there is a recruitment consideration and patient availability to participate in the clinical trial. For us, we are excited about running the trial both in US and ex-US. I only see advantages of doing this. Yeah, we are planning to be global. So there are some particularities. The regulatory clearance takes a little bit longer, ex-US, but the sites are amazing, both in US and ex-US. So we are hopeful to be collaborating with both parts.
And I guess I would add, like, in addition to, in terms of other available treatments, for different genetic forms of ALS, the frequency of the genetic mutations varies across regions. So, for example, SOD1 ALS is more common in people of Asian ancestry. So you would be more likely to find those patients in, that region of the world compared to, you know, Europe or North America. Um, I think at this point, both across Europe and across North America, they have very kind of, developed consortia. So, the great networks of sites, highly experienced. As Olga mentioned, the regulatory peculiarities are a little different between the two, but overall, really, really strong sites. We've had, an incredibly positive experience working with Canada for our SOD1 studies. So that's been a very good experience for us as well.
Yeah, I think in ALS, it's really unique situation where, in all parts of the world, the clinical sites are doing their best to initiate ALS trials as fast as possible. They understand the urgency the patients have, and it is a truly inspiring collaboration we have so far.
Thank you both. I've got one more for both of you. Before we have a couple more questions for you, Olga, as well. Both of your trials are using different strategies to target specific biology. One is siRNA, the other AAV vectors. Can you talk about the differences to these strategies?
I guess
Well,
I can start.
Go ahead. Yeah.
So, the siRNA, it's clearly designed when you want to reduce the levels of something. So if your approach is you have something and you just want to reduce its levels as strongly as possible, siRNA is probably a pretty good choice. Olga's approach allows you to be maybe a little bit more nuanced in terms of what you're doing, but in terms of that, siRNA is a good choice. I think another consideration is, do you want to be a one and done treatment where you're able to give a single administration and then, you're done, or you need to give ongoing administration? In some ways, we think about siRNA as maybe something in between, right? If you're dosing, once or twice a year, that is something between, let's say, a monthly intrathecal dose or even, a daily dosing of an oral medication and a one and done. So again, it really kind of, depends on the specifics.
Yeah, I think, Oren, you covered it. Just maybe to add that in our case, AAV is just a delivery vehicle, but we know that, and as I just presented, TDP-43 is a crucial protein, so we cannot decrease the TDP-43 level, the normal TDP-43 level. So this is why our antibody is designed to be highly specific to target pathologically misfolded TDP-43. But in the case of SOD1, it is a difference. So it really, the choice of the treatment modality is really driven by the biology, and it is not, so it should be fit for purpose.
Thank you.
All right, this next question is for Olga. So this first question was partially addressed in the presentation, which was, is there a marker for TDP-43? But then, if not, how is it known that 97% of ALS patients have abnormal TDP-43 pathology?
Neuropathological studies, so postmortem analysis.
Got it. Thank you.
That's kind of funny. Thank you. So it appeared from someone in the audience that the recent Neal's annual meeting that ALS research is now focused more on gene therapies rather than pharmacology or small molecules. Is that accurate to you as you read the landscape and how it's evolving?
I think we are moving towards the era of more precise medicine approaches. I think the ALS scientific community is just capitalizing on the success of the person. And also there are new emerging treatment modalities like Oren presented, like ours. There are much more treatment modalities out there as well, which I don't think that there is much more focus. I think the community still wants to try everything which makes sense biologically. But probably the probability of success in the genetic subtypes is higher.
Yeah, so I would add exactly that in the genetic subtypes, it's pretty clear what's the underlying driver of the disease. So you know exactly what you should be targeting. And then I think the other way you can think about like genetic approaches is basically using, the value of the specificity of genetics to very specifically target the protein that you're looking for. So both in terms of genetic causes and also genetic approaches to be more specific in terms of targeting what you're looking for. There probably is a move towards using both of those, but I wouldn't say that it's exclusive. I think there's still a lot of work using more traditional pharmaceuticals in the space.
Thank you.
These questions for Olga. What restrictions or exclusion criteria are going to be in place for the clinical trial? Maybe SVC or FVC, ALS, FRS score, time since onset.
Yeah, we are at this point, I cannot disclose the full set of eligibility criteria, but hopefully early next year we'll be able to rediscuss this. We are planning to select patients based on a specific duration since symptom onset and specific percentage of SVC, indeed. And I think it's important to mention as well that although our approach is a vectorized antibody technology, we fall under gene therapy bucket regulatory-wise. So we have to follow patients for a long time, and we need to ensure that we include the patients who are able to undergo study procedures and remain in the study for a reasonable period of time.
Thank you.
Yeah, actually, I'm going to go off script from the initial questions and follow up with something because it's both what Olga, what you just explained, and Oren, what you were talking about in terms of precision medicine. So these types of medicines and these trials do refer more to either one and done or a few doses. That means a lot of monitoring in between. Do you see that there's a good amount of chance for there to be more kind of creation of data from the kinds of studies we're doing to create more subsets of patients that can participate in studies and really target people and filter them the right way for trials to match them? Is there an urgency there for you to be a part of kind of that kind of work where patients more is known about them on the way in, and you can actually maybe generate more precise therapy plans for them that way, using like gene therapies and related precision medicines?
So just to ensure I understand correctly, Christian, are you referring to pre-treatment disease trajectories?
Yeah, exactly.
Yeah, I think it is. It's not necessarily only for gene therapy or siRNA or ASO. It is just important in general. Any effort and platforms which help to well characterize and define a trial-ready cohort with some amount of pretreatment clinical data and hopefully biomarker data will be crucial for success, I think. Oren.
Yeah, totally agree with that. And then for, genetic ALS, it's a challenge to identify patients who are carrying SOD1 mutations. A fair number of them have a family history, but probably if you look at the numbers, probably about half of the SOD1 ALS patients do not have a family history and may not be getting genetically sequenced. I think now that tofersen is on the market, I think most people are at least getting offered genetic sequencing. But still, it is like a potential bottleneck, especially, if people are thinking about other monogenic forms of ALS, finding the patients can be a real challenge.
Thank you.
There are a couple questions about looking at other diseases and how they can kind of inform ALS research. So one was, about cystic fibrosis and how the discovery of the gene led to 3 specific drugs to give to cystic fibrosis patients. And then also in oncology, the use of multiple compounds is common. So one, how do you think of, looking at other diseases as a model for ALS research, and how far do you think we are from, doing things like looking at a particular gene or maybe using multiple compounds?
Well, I think this work is already ongoing, and I think we should start looking at the lessons learned from other neurodegenerative disorders first, and it will be obvious when looking back into what happened in the Alzheimer's world that the success, occurred only once the field had a set of very well validated biomarkers, and which helped to well define the patient population when the diagnostic paradigm shifted to the left, allowing to diagnose earlier. But there are other lessons learned, obviously. I think that these are great suggestions to look also beyond just neurodegenerative field. And and we were just discussing the genetic mutations where we may have a higher probability of success. It depends obviously on the mutation. We had some disappointments with C9orf, but we also had great success with SOD1. So yeah, Oren,
Yeah, in terms of the genetics, it's, SOD1 was, I think, discovered in the early 90s, and it took a long time to figure out how to, therapeutically address it. Huntington's disease is a similar story where there is a single, mutation that causes disease in all the patients. And it's taken a very long time to try and wrap our heads around what the best way to kind of therapeutically intervene. So even in the quote unquote simple cases where there's a single gene mutation that's causing the disease, it can take a long time to figure out how to get the therapy right in order to make a difference. And then regarding combination therapy, I mean, intuitively, it makes a lot of sense, right? I think it's challenging from, kind of the drug development perspective. It is extra work because, from a regulatory perspective, you need to understand what each thing is doing on its own and then what it's doing in combination. It just makes the development a lot more complicated, especially when each thing that you're trying is something novel. So it's, there is a little bit of an inherent challenge and complication in terms of trying to develop combination therapy. Intuitively, it makes a lot of sense, but it's a tough road.
Thanks. So exactly along that line of, the complexities are more than just what they're more complex than we think, even though there's a single gene that we're targeting. When do you think we'd be able to predict the use of siRNA for sporadic ALS? How would we be able to start to test and untangle the gene sequencing aspects? And how would this information be interpreted to be useful?
I think it's all about finding the right target. So, as Olga mentioned, TDP-43 is clearly intimately linked with, ALS in most patients, but you can't use siRNA to target it because if you just reduce the levels of TDP-43, that's toxic. So it's all about finding the right target where the goal is to reduce levels of that target. And if you think you can do that in a safe way, then developing an siRNA that tries to reduce that target protein is a great approach. So I think that's the challenge is, do we have targets where just clearly knocking it down is the right way to therapeutically intervene? If we have something like that, then I think siRNA is one amongst many of modalities that could potentially be useful.
Sorry.
All right, this one's for Olga. Is it true that VTX-002 is being developed to modify the disease and not just manage symptoms?
yes, it is being developed as a disease modifying strategy. Just true.
Great, thank you.
And we've gotten through a lot of the questions. I think one good question that we had ready for you was, what do you think the biggest learnings are from past ALS trials, and that you've found yourselves applying, and that you think are helping your designs or your approaches to your trials?
I think in the SOD1 space, I alluded to it before, the power of the biomarkers that tell us about how much SOD1 is being expressed, and the power of neurofilament light protein to be able to tell us, are we making a difference in the patient's kind of, disease activity? These are incredibly powerful biomarkers that really can give us a hint in a relatively small number of patients that we're on the right track. So that ability to be able to run an early study and not just, try and figure out what dose of your compound is safe, but actually get information about whether or not you're on the right track in affecting the person's kind of disease activity and disease trajectory, incredibly powerful. So I think for us in the SOD1 world, that's huge.
Yeah, I think we are taking advantage of lessons learned from both failed and successful trials, both in sporadic and genetic forms. I think the field in general learns a lot from the first experience, although it is a genetic subtype, but a lot of lessons learned can be translated into larger ALS patient population. So I think the major ones are the window of intervention. Another one is how long you need to wait to see the clinical benefit, and that the biomarker response may come sooner than the clinical response. So you need to give a chance to your drug, to demonstrate its effect. Yeah, I think these are critical things, and obviously biomarkers and NFL and beyond.
Are your companies thinking about using technology to collect data from patients at home during trials to make enrollment maybe more scalable and more accessible?
We are very interested in using wearable sensors to help us understand patient function. And part of that obviously would be kind of collecting data at home. I think, that's where our greatest area of interest is. We think that type of approach can give us a very complementary set of information to the way patients are functioning compared to the usual ways we measure that in terms of, asking patients how they're doing during site visits or during remote calls.
Yeah, we are obviously assessing digital technologies as well. For our early first in human study, the goal is to define the safety profile and to understand some early biomarker signals. But as we move forward with the clinical development, obviously this is one of the things which is on our radar.
Oh, actually, one question that I almost missed was, do your programs, are they amenable to EAPs? Do you think you plan to offer EAPs as you go along?
I think we certainly want to be able to kind of provide access as much as possible. I think, we'll wait to see, are you seeing clear signs of activity? A lot of it also depends on the status of the clinical trials we're running. Are they still active, or have they, kind of closed recruitment? So kind of within that context, we would, obviously want to be able to, once we're convinced that the drug, has, potential benefit and shows safety. If there's not availability of a clinical trial for a patient, then we would certainly consider kind of providing access through a program like that.
Same approach here.
Thank you. We have gone well over tonight, I know. Thank you, everybody, for all your questions. I think we got through almost all of them. And if we didn't get to them, then we will certainly pass them along to Olga and Oren for them to be able to provide their responses to you. I want to thank our guests tonight. Like I mentioned at the very beginning, both of our guests tonight are incredible champions of what we're doing here, and really supportive of the kind of data that we're collecting for everybody who participates in our studies. remote studies. They're big believers in the power of the kind of insights that we can generate that support the trials and eventually get these new treatments to market. So thank you again, Olga and Oren. I hope, McFinn, I turn it back to you. I hope it was a valuable program for you as well.
Thank you for
Sure.
inviting us.
Yes.
Thank you.
Thank you.
We're extremely grateful for your time, for your experience, and everything that you have collected up until today. We're looking forward to the new things that you're going to find out, and we hope that you come back with some exciting, what do you call that? Hope. That's right. So it's all made from all of us, for you two doctors, for us who support you, and for the people with ALS. You're very important. So please go home, get some good sleep, and spoil yourself some way, please. Folks, the day goes by pretty quickly, and our programs go by pretty quickly. I wish they would slow down so we could spend more time together, but that's why we have a program constantly. So for all of you who have joined us tonight, you have created a larger family that you are part of. So always remember, when you have something that is a dire need, you can always contact a Pathfinder and see maybe they just might have the right answer for you. And with that, I want to celebrate that each of you came here tonight, and each of you probably saw someone in another window there that you might like to say goodnight to. So on 3, let's everybody find that person that you want to say goodnight to. 1, 2, 3.
Good night, all.
Good
night, everybody.
Dr.
Dr.
Good night, Linda and Oren.
Good night, good night, Shirley. Good night, Penny. Good night,
John.
Olga.
Christian.
Good night,
Good night,
Todd.
Tony.
Good night, Tony.
Good night,
Good
Pat
night,
Mon. Good night,
John.
Dwayne.
Good night,
Carl,
Olga.
thank you so much.
Good night, Lyle. Thank you again.
Big
Come
fan.
on, there you
Olga
got
and
it. Come
Oren,
on out of
thank
there, everybody.
My favorite
Good
Good
night,
night, Amo.
Oren. Good night,
Wow.
Good
Oren.
night, Eddie.
Debbie. Good night. See you all
Thank you
next
so
time.
much.
Good night,
Thank
Tony. Good
Good
night,
night,
Sherry.
Steve.
Good night, McFinn.
Good night,
Sherry,
Mary and
good
Deborah.
night.
Good night.
Thank you for
Richard.
taking good care of Tony.
Yes.
All right,
Good night,
Pat
Carl.
Munn. Good night, Pat.
Good night,
All
John.
right, folks. Hey, Joe Redmond, keep those hearts
Can't
popping.
see your close up. Oh well.
All right. Love you all.
Yeah.
Looking forward to
Love you
seeing you
guys.
all
Thank
soon.
you.
Be well.
Just know that love is possible and hope is medicine. All right.
Have a
Good
great
night,
day.
everyone.
Okay.
Sleep well.
Next time.
Yes, next time.
Time.
Bye. Bye, Carl.